Hi and Assalamualaikum to Dr. Yaya. I'm going to be the first member of the group 2 that will present first. Let me introduce myself. My name is Zulaika binti Muhammad Najib. My matrix number is F22B0037. So today I'm going to present about functional study of central nervous system for farm animal husbandry. But first, did you know that animals are taught to categorize whether sensory stimuli are positive, rewarding, or negative? Understanding the central nervous system in the context of farm animal husbandry and study about the neuronal mechanism underlying learning and memory are required in order to understand behavioral development in farm animal. To understand it more better, I will pass it to the next member and she will talk about the purpose of the study of functional study of central nervous system for farm animal husbandry. That's all from me.
Hi, my name is Lim Xiaowei. My true number is F2280102. Now I'm going to present the purposes of the study. First, the purposes of the study is to examine the role of the central nervous system in regulating physiological processes and behaviors in farm animals. Second is to investigate the influence of environmental factors on the functioning of the central nervous system in farm animals. Third, the purpose of the study is to explore potential strategies for optimizing farm animal husbandry practices, improving animal welfare, productivity, and overall management. That's all for me. Thank you.
Results and discussion. Hi, my name is Shukrina Aisha binti Fakurazi. My matrix number is F22A0112. Olfaction. Methodology using the electrophysiological recording technique with microdialysis. The mitral cells in the olfactory bulb respond to lamb odors only after parturition. Gamma aminobutyric acid and glutamate are released in response to the odor of the sheep's own lamb, while acetylcholine and norepinephrine do not have a specific response. The olfactory discriminating ability of lambs may be weak in the case of caesarean operation, suggesting a need for further studies on farm animals to investigate perception, learning, and memory of natural odorants. Taste. Methodology. Conditioned taste aversion involved. Conditioned taste aversion involves pairing conditioning and an injection of lithium chloride. Animals that experience conditioned taste aversion refuse certain kinds of tasks after the combination of conditioning and lithium. The retention of taste aversion learning and memory is long-lasting, as the aversion can occur even when the cue and aversive experience are separated by many hours. Conditioned taste aversion is used to train cattle in the field to avoid eating toxic grass.
Oh, hi and Assalamualaikum. My name is Nur Anis Elia binti Nur Hasmadi. My matric number is F22A0291. So I'm going to present about hearing and vision. So as for hearing, cattle, goats and pigs has been found to be less sensitive to change in the position of sound sources compared to human and dogs. So my first point of view is in cattle and goats, the threshold for behavioral responses to change in sound source position along the medial sagittal plane is between 18 to 30 degrees. Meanwhile, in humans, the threshold for behavioral responses to change in sound sources position is 0.8 degrees, while in dog is 8 degrees. But for pigs, they have better hearing than humans, which is their hearing is more sensitive than humans actually. So these findings suggest that the hearing modality difference between species. So as for vision, ruminants have dichromatic color vision with peak sensitivity in the range of 444 to 455 nanometers and 552 to 555 nanometers, which means bulls have a visual acuity that allows them to identify a white center as small as 1 centimeter of a black disc. Bulls' visual acuity also decreases with aging compared to sheep and younger cattle. Bulls perform better in discriminating vertical stimuli compared to horizontal stimuli in an operant conditioning task. Meanwhile, sheep have the ability to casually recognize humans and perceive and perceive differences in movement and clothing. So that's all from me. Thank you.
Hi, my name is Teo Cui Shan and my matrix number is F22A0120. So now it's my turn to present the content about neuroendocrinological studies and stress-induced behavioral changes, the results, and also the discussion for this part. So there are few tests and also few experiments conducted, and those animal involved for those tests and experiments are both different. For example, like there are domestic pigs, cows, and also cattle involved in different tests. I will start explaining about the insights from those tests and also experiments conducted. So the first thing is about the activity of the autonomic nervous systems, which is influenced by amygdala and also hypothalamus. It is important in regulating the heart rate, blood pressure, and also hormonal secretion. And next thing is about the CRH, which is the corticotropin releasing hormone. It's the key hormone that integrating the stress response. And then next, about the test of the separation also reunion in cattle suggests that the genetic background and also central pathways involved in influencing the animal's sensitivity to stressor. And the next thing is about the shape of circadian rhythm in cortisol concentration has been proposed as the key parameter in assessing chronic stress and also welfare in animals. Next thing I would like to explain about the stereotaxic approaches in the brain of farm animals. So as you can see through the picture there, stereotaxic instruments is something like that, and involving using also a brain atlas to analyze the regulatory role of the intracerebral nuclei and in the systemic regulation. So the stereotaxic instruments or the stereotaxic approaches is still in developing stages. So for some of like larger size animal, they didn't have the approaches to these instruments yet, but everything is like in stage of developing.
Assalamualaikum, my name is Adina binti Muhammad Tamizi. Number matric F22A0191. Okay, for the conclusion, the functional study of the central nervous system (CNS) for farm animal husbandry reveals the important role it plays in regulating psychological and behavioral processes in field animals. Firstly, we have explained how the central nervous system is involved in regulating vital psychological functions such as sensory perception, motor control, and the regulation of essential bodily processes. The central nervous system allows farm animals to perceive and respond to the environment, control voluntary and involuntary movement, and maintain homeostasis. Secondly, by improving animal welfare strategies and management practices based on the knowledge of the CNS, we can enhance the well-being of farm animals. Adequate housing, environmental enrichment, proper handling, and access to veterinary care are essential components of promoting animal welfare. Lastly, integrating the functional study of the CNS into livestock farming contributes to the overall well-being, productivity, and sustainable development of the industry. When farm animals experience better welfare, their productivity increases, resulting in higher quality product. By recognizing the role of the CNS in farm animal husbandry, implementing improvement management practice, and prioritizing animal welfare, we can achieve a balance between the well-being of animals, productivity, and long-term sustainability. This benefits farm, consumer, and the agricultural industry as a whole, promoting responsible and thriving livestock farming sector. That's all. Thank you.